All right. I think just in the interest of time, I think I'll go ahead and start the introduction. And if people are joining, I think they can, it'll still work out. So, thank you everyone, first of all, for joining the session today. This is a combined event of the Firefighter Cancer Cohort Study and the Center for Firefighter Health Collaborative Research, the latter being at the University of Arizona, and the first being kind of a national collaborative effort. And in that regard, it's a pleasure to introduce to you Dr. Heather Stapleton, who is a professor, environmental chemist, and exposure scientist in the Duke University Nicholas School of the Environment. Her research interests focus on identification of halogenated and organophosphate-based chemicals in building materials, furnishings, and consumer products, and estimation of human exposure, particularly in pregnant women, children, and firefighters. And she'll obviously be talking a lot about firefighters today. She is an internationally recognized expert on exposure monitoring using silicone wristbands, and I consider her the leading expert in this field and others. She currently serves as the director for the Duke University Superfund Research Center, and Heather is also a co-lead with the Firefighter Cancer Cohort Study and is enrolling North Carolina firefighters into the FFCCS as part of a larger PFAS study that's funded by their state. So with that, I really appreciate the fact that Heather's willing to share her presentation and expertise with us today. We're setting this up so that hopefully we'll have plenty of time for questions from all of you, and we're recording this too, in case someone would like to refer to it later. So with that, I'll pass it off to you, Heather. Thank you.
No, great. Thank you, Jeff. We appreciate the opportunity to chat. Good afternoon, everyone, or for anyone out on the West Coast, good morning. Let me just bring up my slides again. Jeff, can you confirm, is it in the right version or does it need to be in the other version?
Other
Does it look
version,
like it's
other version.
okay? Thank you. I have
Mhm.
two monitors, so sometimes
There
it's
you
very
go.
difficult
That's
for me
right.
to. Thank you very much. Well, just like good afternoon, everyone. As Jeff indicated, I'm an exposure scientist here at the Nicholas School of the Environment at Duke University. I've been here for 20 years. And we've been using silicone wristbands as an exposure tool for the last 10 years. And what I wanted to do today was take some time about talking a little bit about how wristbands work and how we're applying them to answer some important questions within the fire service. So I don't think I have to tell anyone why we're concerned about chemical exposures in firefighting. Obviously, we know there are health risks associated with firefighting, both short-term respiratory problems and long-term risk for long-term diseases such as COPD, cardiovascular disease, and unfortunately cancer. As Jeff already alluded, myself and Jeff are part of a firefighter cancer cohort study, and this is obviously on all of our minds, trying to understand what the drivers are for cancer risk in the fire service. And as you can see here, on average, there's some statistics suggesting it's 14% higher in firefighters relative to the general population, but obviously that will vary depending on the specific type of cancers. In 2022, the International Agency for Research on Cancer classified the occupation itself of firefighting as a group 1 carcinogen, which is the highest level that they designate, because there was sufficient evidence, particularly for mesothelioma and bladder cancer, and then some more limited suggestive evidence for a few other cancers you can see here on the left of this figure. If you look over to the right of this figure, obviously we know that there's some different stressors and exposures that firefighters receive, including exposure to combustion byproducts, diesel exhaust, building materials, asbestos, chemicals, shift work, UV radiation, and that can vary over time depending on the different activities that a firefighter participates in. And it can also vary depending on the type of fire or the response. There's exposures are going to be different in structural fires versus wildland fires versus vehicle fires. And then we're going to talk a little bit about that today and why it's important to understand that. So if we want to do something about this, right? We want to elicit change. We want to help inform policy decisions that can reduce these health risks. we need to know where these exposures, what exposures are driving this, what are of most concern, where do they originate, and are there specific activities that are influencing the overall exposure to these chemicals? Now, we have a lot of research that's already been generated on exposure to these chemicals. There's some things we know and there's some things we don't know. Typically, the standard approaches for assessing chemical exposure are through collection and analysis of blood and urine. And these are well established, and obviously they're the gold standard. There are some limitations to these, and that's what I wanted to mention, and where kind of wristbands and wearable samplers can play a role. Now, typically we collect blood and analyze blood for chemicals that have what we call long half-lives in blood. And some chemicals that have long half-lives are things like PFAS or the pesticide DDT. Because once we're exposed to them and they get into our body, they stick around for years before they're broken down or excreted by our tissues. So what you measure in blood is actually a very good marker of your long-term exposure. But other chemicals are rapidly metabolized or excreted in our body. And typically, we will focus on urine as a sample for assessing exposure to those chemicals because they're broken down and typically excreted in the urine. So some examples are things like nicotine or combustion byproducts, particularly polycyclic aromatic hydrocarbons, or what we call PAHs. But as I said, there can be limitations of these approaches. First of all, it can be challenging to collect blood and urine, particularly in the field under certain circumstances. Really hard to get a phlebotomist out there to draw a tube of blood. If we're looking at different chemical classes, sometimes you need multiple tubes of blood, right? So there's some limitations there. But even if we're specifically talking about certain chemicals, like taking PFAS, for example, as I said before, they have a long half-life. So what you measure reflects long-term exposure, right? So it's more challenging to identify what your short-term exposure to PFAS is if you're interested in understanding, was I exposed to PFAS when I responded to this one call or over the last few days? Because there's been so much accumulated in our body from long-term exposure that's hard to differentiate that. In addition, we know that PFAS, we receive exposure from our diet, particularly drinking water in some areas. So when we measure this in blood, we cannot tell how much of that came from our diet versus how much of that might have come from an exposure that we experienced from a fire call. So that becomes a potential limitation. And even for things like urine, as I mentioned before, these are chemicals that are rapidly metabolized. So if we take a sample in the morning and measure combustion byproducts, and we take a sample in the afternoon, those levels can be very different because it depends on when you're exposed and how fast your body metabolizes this. And actually, different people can metabolize things at different rates or sometimes to different metabolites. That's rare, but it can occur. And when we measure these metabolites or breakdown products in the urine, there are some chemicals, and PAHs and plastic additives are included in that class, in which the metabolite is not always specific to a parent compound, because you can have multiple chemicals that break down or metabolize to the same metabolite or breakdown product. So it can be difficult to tie that exposure back to a specific parent compound. And just like in the case of PFAS, these exposures can also come from the diet. PAHs we know originate from combustions, from fires, but you can also get exposure to PAHs from certain food items, particularly if you had something smoked. Like here in North Carolina, we have a lot of pulled pork where you're having smoked meat products. You can also have higher exposure. So again, when you measure that urine, you can't always tell how much came from your diet versus how much came from that recent exposure at a fire call. So I'm not saying we shouldn't collect blood and urine. I absolutely think we should. I just think we need to complement them, right? So thinking about how we can track these exposures over defined periods of time to better understand these exposures or based on specific job tasks or activities. And in my mind, we need to kind of use these wearable samplers to get better measurements of exposure for specific individuals because your exposure will change if you're closer to a fire versus farther away. So having that individual level precision is very important. And we need something like a radiation badge that's picking up that exposure from the ambient environment. And this is where I think silicone wristbands can help. They have limitations as well, but I think they're a good tool and they have questions they can ask and answer that are not always feasible using blood and urine, right? So let's just talk a little bit about the silicone wristband. There are chemicals that are all around us in our air, thousands of chemicals. Often some of those are really important. They're helpful for us, obviously oxygen, but then we have other chemicals like the combustion byproducts. some just other releases from emissions, things that come off building materials. These chemicals are in the air. We can inhale them. They can sometimes stick to very small particles that we can't always see in the air. Those particles can settle on our skin. They can attach to our clothes. And then we can be absorbed to these chemicals through these three different routes, right? Inhalation, ingestion, or dermal absorption. And wristbands do a really good job of measuring exposure to organic chemicals. I should mention that we're not using these for metals. There's some limitations with metals, but we are working on that. For organic chemicals in which inhalation and dermal exposure are the predominant routes of exposure. So it's something we wear in this case, on our wrist. So they're not capturing those exposures that occur through the diet, through what's in our drinking water, or what's in our food. And in this case, I think that's an advantage if we're really wanting to understand exposures that are occurring in the workplace, right? And in both the case of inhalation and dermal, there's this contact between the chemical and an interface on our body, right? So when we're inhaling air, which has chemicals in it, it enters into our lungs. And then in the lung, it's crossing an epithelium to enter our bloodstream. And on the dermis, right, these chemicals can come into contact with our skin, and then they'll also cross the skin barrier, the stratum corneum, and enter circulation. Wristbands kind of act as that barrier, whether it's in the lung or in the skin, and chemicals can stick to it and then absorbed into the interior of the silicone. So in that way, it mimics some of the absorption that occurs across these boundaries in our lungs and on our skin. So it's a good surrogate for measuring exposure through inhalation and dermal. And in this case, does not capture exposure that occurs through the diet. We know from other studies that we conducted that silicone wristbands can accumulate these chemicals over time. So the longer they wear it, the more that it'll accumulate. So this is highlighted by the graph on the left. So even though we're taking showers when they might get wet, a lot of chemicals don't wash off. They will stick to it and they just accumulate over time, which is why it's important for us to track how long someone wears this to take that into account. So this offers some advantages over these traditional measurements like a urine and blood collection. So the Venn diagram on the right is trying to highlight some of these because it can isolate those inhalation and dermal exposure pathways and rule out dietary exposure. They're really just capturing exposures that occur while you wear them. And then these are pretty stable. We actually mail these back and forth to different groups and populations, and the chemicals are fairly stable on these wristbands up to a matter of weeks. We certainly put them in the freezer when we get them back, but that's one advantage. And they're easy, right? We can put them on, we can wear them, and then we just take them off when we're done. So they have a lot of advantages to supporting exposure scientists. They also go everywhere we go, right? So in some cases, we might be interested in our exposure when we go to different places or different microenvironments, right? So this is a generic slide I like to use. They can capture exposures that happen in the home, but if we get in the car and we travel somewhere, they're monitoring those exposures. Or maybe we're interested in exposure at work, and wristbands will integrate these exposures over these different microenvironments and over time. But in this case, maybe we're just interested in exposure in one place, right? Occupational exposures, or in this case, exposures experienced by the fire service. So we just wear them while we're working, and that helps us understand this exposure. So that's why we are interested in using wristbands in these studies to answer some of these, ask and answer, hopefully, some of these really important questions, right? We actually started our first project working with firefighters here in North Carolina was in 2019, and I'll talk a little bit about that one. It was a very small scale study where we just started with the basic question, how do the chemical exposures differ when you're on duty versus off duty among certain firefighters here, structural firefighters? That worked really well. We learned a lot, and we wanted to expand and move beyond those kind of questions. Are there different exposures that we experience when we're responding to a structural fire versus a vehicle fire? Are our exposures different depending on what kind of job tasks we perform while responding to these fires? And lastly, and we'll talk a little bit about this today, are these chemical exposures different depending on how often we clean our gear? I think that's an important question and hopefully something actionable we can do. So I'm just going to take a few minutes to go over how we quickly prep and analyze the wristbands and then jump into some of the studies that are ongoing and talking a little bit about what we've learned. So here's just a picture of what we call a wristband kit on the left. It's this wristband that we actually clean in the laboratory. We actually buy these in bulk from a manufacturer of wristbands. And these are just standard polydimethylsiloxane, PDMS. They are the same type of wristbands my kids get at fundraisers. They're very common. We buy them for 10 cents a piece. But they do have chemicals on them when we receive them. So that's why it's really important to clean them in the lab, and we keep them in these airtight containers, these aluminum tins that you see here. Then we deploy them. So we ask participants to wear them for a specific amount of time and record that time. As you can see on the photo on the left, there's a space where we record the date and time they put it on and the date and time they take it off, because that's also really important. As I said, exposure changes over time, so we need to know how long someone has worn this. They are pretty stable for a couple weeks. We can mail these back and forth, but when we do get them back to the laboratory, we do put them in a standard freezer until we analyze them. I won't go into a lot of analytical details here, but just in case people are interested, what we do in the lab, we actually just take a very small piece of that wristband. It's about a centimeter. For analysis of all these chemicals. The rest stays in the freezer in case we ever want to go back and look at something else, because we don't need a lot. We don't need that whole wristband to make these exposure measurements. We take a small piece, we put it in a test tube with some solvent, and we actually use sound waves, high energy sound waves in a sonicator, and that vibrates the silicone, and these chemicals come off into the solvent. And we can take that solvent into this little vial, and then we use what we call mass spectrometry to quantitate how much is in that wristband and what that particular individual was exposed to. We actually have two approaches to analyzing these silicone wristbands and their extracts. The first is when we look for specific chemicals, and that's when we say it's targeted. We're telling an instrument, look for this specific chemical with this mass, and we actually run an example of that chemical on the instrument at the same time, so an authentic standard for comparison. So we have about 170 chemicals that we quantify through this approach, which includes combustion byproducts, some PFAS, flame retardants, which are added to a lot of building materials and electronics, pesticides, other plastic additives, and some industrial chemicals. But we also have the ability to do what we call non-target analysis. Are there chemicals on that wristband that we weren't looking for? And we do have the ability to do that. Now, today I'm only talking about targeted analysis because we're very confident in that. The non-targeted aspect will take a little bit more development, but it is in process, and we are learning a lot from that. So what I was going to do today is talk about two studies, one focusing on structural firefighters here in Durham, kind of these small pilot studies, and I'm going to spend some more time talking about some data so we've collected in collaboration with some wildland firefighters in Utah through the FFCCS. I'll first start with this study we conducted. This is the first study we did back in 2019, and this was supported by my former PhD student, Jessica Lavasser. This is now published. In this case, we worked with structural firefighters in Durham, North Carolina, and asked them to wear wristbands during different periods of time. Wear them while they're working, and then record whether or not you respond to a fire, but also wear a new wristband while you're not working, while you're off duty. So each of our firefighters in our study wore a silicone wristband at these three different time points. So we had three different wristbands from each of the firefighter. So we could look at the exposures and how they changed between these different time periods, off duty versus on duty, and then when they're on duty, whether or not they responded to a fire. I'm just going to first kind of comment on what we found, what were the take-home points, and then I can certainly show the data. On average, what we found is that exposure to combustion byproducts, these polycyclic aromatic hydrocarbons, were significantly higher when responding to a fire call compared to off-duty. Probably no surprise, we've seen that in other studies. What was also interesting to me is that we saw that these exposures to these combustion byproducts were also elevated even if they didn't respond to a fire. So they're working, they're performing other activities around the fire station, but they were higher than when they were off duty. And why is that? Is that something about exposures in the fire station, the materials, training? It really wasn't clear to us. It was the first study, but these are additional questions that we'd like to ask and answer. But we also saw that there are some chemicals that were lower on duty, right? Certain pesticides, certain chemicals that are used in some consumer products. So not everything goes up, some things go down when we're working, which was a nice proof of concept. Here's some of the data that's actually published in the figure if people are in the paper, if people are interested in looking at it. These are what we call results from a regression analysis, where basically all these different combustion byproducts and their very long names are on the bottom here. These are all PAHs. And then the scale on the left here on the y-axis is looking at the multiplicative change in the average exposure to these compounds, either on duty versus off duty. So basically, we're looking at that line labeled 1. So it says off duty, that's our reference, right? And looking at how much our exposure changes. Is it higher? Then it's going to be above that line. If it's lower, it's going to be below that line. And what you can see here is most of these PAHs were higher on duty versus off duty, particularly when responding to a fire, which are the symbols, the square symbols here. But you'll notice that the circles are also above the one line, and that means their exposure was also higher on duty, even if they didn't respond to a fire. Why is that? And that's an important question I think we need to ask in the future. In addition, we looked, as I said, other things. These are phthalates, which are plastic additives. Some of them are used in personal care products. Here we saw most of them didn't change on duty versus off duty, but there were a few that were actually lower when firefighters were working. And these are actually what we call low molecular weight phthalates, which are more commonly used in personal care products. So things like lotions and in things like shampoos and other things. So we think maybe this reflects differences in personal care product use on duty versus off duty, like what you have in the station versus what you have at home. Because we saw the same trend for some pesticides that are used in flea and tick medications, probably because some of these exposures are related to use on pets that were at home but were not at the fire station. So we saw these kind of really interesting patterns. We did continue to work with the Durham firefighters and assess their exposure to PFAS with silicone wristbands. This is the second publication that is now out and published. I'm just going to briefly talk about it because I know there's a lot of interest in PFAS. This was also another small pilot scale study with 23 structural firefighters where we asked them to wear a wristband, a new wristband for 9 days over their shift because they're on 24 hours and then off 24 hours. And then they'd be off duty for 5 days and then they come back again. And so when they came back for another 9 day shift, put a new wristband on. And they would repeat this over 4 to 5 months. And at the end of the 4 to 5 months, we collected a blood sample. So we were curious about exposures on the wristband over time and how it related to PFAS at the end of that period. Now, it's important to note we looked at PFAS and we've also looked at other chemicals. What we measured for PFAS, and we could detect them on the wristbands, was generally pretty low, much lower than what we see for combustion byproducts, flame retardants, and even pesticides. But they are there, and that's probably not surprising to me, I think, and others, because we know a lot of our exposure does come from our diet. But we did see one PFAS, which I have in the plot on the right. It's called methylphoce. It was significantly correlated with its breakdown products. So not all PFAS are forever chemicals. There are some that break down. In this case, methylphoce breaks down into a chemical called N-methylphosa. It was significantly correlated between levels on the wristband and levels in the blood of these firefighters. And so we see this clear trend, which is good. It reinforces that the wristband is actually measuring meaningful exposures for these compounds. And it's important to note we did observe this similar trend in a non-firefighter cohort. Methylphoce is a common PFAS that has historically been used in furnishings, so as a stain repellent on upholstery and furniture, for example. So we believe this stems from certain products in the home, and it's kind of ubiquitous in the general population. So it's not necessarily specific to firefighting. There was another PFAS, I don't have this plotted here, that goes by the acronym MEFBSE. This was correlated with time spent at fire. And this is a PFAS that we know has been found in the moisture barriers of turnout gear. This is something we're also interested in following up. So that exposure could be from responding to fire events because it is also in certain building materials can be released, or it could be from wearing the turnout gear. We don't really know. And it was also a very low exposure. It is not one of the PFAS that has health concerns associated with it. And we actually don't know a lot about its health concerns right now, but we did detect it and we did measure it. So what I wanted to do now is switch to wildland firefighters because we have more data from some recent studies using wristbands within wildland firefighters. So in this case, we asked wildland firefighters to wear a wristband for a certain shift, which is usually on average about 16 hours. So this is working with firefighters out in Utah. They would wear a wristband for their shift, come back, take it off, and also provide a urine sample at that time, and also complete a brief survey just talking about activities that they were involved in while wearing that wristband. So the data I'm about to show you is from 55 wildland firefighters. Most were male. We had 2 females in the group with an age range of 18 to 59. On average, they wore them for 15 hours and 40 minutes, but there was variability there. So all the data has been normalized to the time that these wristbands were worn before we performed any statistical analyses. So as I mentioned before, we look for a lot of different chemicals on these wristbands, 172 different chemicals in this case. Um, 81 of those were detected in at least one of the wristband samples, and 39 were commonly detected, which means they were found in at least half of the samples that we analyzed. And on the right is just a table talking about all these different chemicals we looked for and how often we found some of these. So we look for 24 combustion byproducts, which are PAHs or dibenzofuran. 10 of those were very commonly detected. We look for PCBs, which are legacy compounds, didn't detect those. We found 2 pesticides, 13 plastic additives of the 43 we looked for. We looked for 51 different PFAS. We only found 2 commonly detected. BFR stands for brominated flame retardants. We looked for 18 of those, 10 of those are commonly detected. And then we have this other category, which includes nicotine, some chemicals in personal care products, and nonylphenol, which is used as a surfactant. So I'm going to phrase this by, or structure this discussion by what was the question we asked and what we learned. So the first question was, are the measurements on these wristbands correlated with levels in urine? That's very important. We've looked at this in the general population, but we know that activities and behaviors can be different in the fire service. So we wanted to make sure, again, that we measure on the wristbands is correlated to what actually gets into the body. So in this case, we had available data for PAH metabolites in urine. So what I'm showing you here are 2 different PAHs, phenanthrene on the left and pyrene on the right, their correlation on the wristband and their metabolite in the urine. So the wristband concentrations, that's a nanogram gram is parts per billion, is on the x-axis, and the metabolite levels are on the y-axis. So you can see in both cases, there's a good, moderately strong correlation between these two, which just reinforces again that what's measured there does represent what's actually being absorbed into the body. So there's one other PAH that we have the ability to look at this relationship. It showed the same thing. So in the interest of time, I'm not putting it up here. We are hoping to look at this relationship for other classes of compounds, but right now we only have data for PAHs to look at this relationship between wristbands and urine. So our second question is, do chemical exposures differ based on job tasks? So when the wildland firefighters came back and filled out that survey, they responded to what type of job tasks they perform. Some were aviation ops, some were line construction. And so we looked at some chemicals based on what they indicated they did during that time. And so here's some data. I'm not going to show all of these 40 chemicals. I'm just picking out a few to kind of and talk about some general trends here. So here I picked out 2 combustion byproducts, phenanthrene and pyrene, which are in the 2 different colors here. 3 of our wildland firefighters indicated they were primarily focused on aviation ops, which is helitack. So there's only 3, so we just have to be careful. It's a small number here. And then 18, their primary job was line construction, 12 indicated mop up, and then there's 14 that had multiple tasks. So, 50% line construction, 50% mop up. Some indicated they were doing what's called hose lay. So there was different tasks that were performed. But what we see is that aviation ops definitely has lower exposure to many of these combustion byproducts, which probably makes sense. They're not as close to the fire itself. In general, line construction and anyone indicating multiple tasks seem to have higher exposure to these combustion byproducts. And this was pretty similar for most of the combustion byproducts. Here's another one, dibenzofuran, where you can see that a little more clearly. In this case, you can see aviation ops. Anyone doing aviation ops or mop-up seem to have lower exposure to this combustion byproduct compared to individuals performing line construction or multiple tasks. But then we looked at something like DEET. DEET is a pesticide that happens to be on our panel, and we look for it. Obviously not something we typically think about when we're fighting fires, but because we have the data, we looked at it. And what was interesting here is maybe no surprise, but people that were working in aviation ops had much lower exposure than individuals working line construction, mop up, or other tasks. Probably makes sense again because they're not in this close proximity to people maybe using DEET or some residual DEET that's in some of the areas where these other wildland firefighters were working. But then there were some chemicals that were actually higher in those 3 people working aviation ops. EHDPP, shown here as ethyl hexyl diphenylphosphate. It's actually a common plastic additive, also sometimes used as a flame retardant, but it's also used in hydraulic fluids. Again, these are 3 people, so we have to interpret this data with a little bit of caution, but the 3 individuals working aviation ops had higher levels on their wristband. They also had higher levels of another phosphate called TBOEP or tris 2-butoxyethyl phosphate, These are both phosphates. They're actually both used in similar ways as plastic additives and also potentially in hydraulic fluids. So this may indicate that this exposure is coming from something in the, I think, in the aircraft itself. And those individuals working other tasks had much lower exposure to this. So again, you can hopefully see how it kind of is differentiating some of these exposures based on the job tasks that these individuals are performing. Another question we wanted to ask is, what about the fuel source? One of the questions that the wildland firefighters answered was, what fuel sources did you encounter during your shift? A lot of people indicated things like trees and shrubs and timber, but we did have a few people that indicated they came across a vehicle fire or a structural fire, which is more like a wildland urban interface or a WUI fire. So here I just pulled out two chemicals. One of these is a flame retardant. One of these is a combustion byproduct to kind of show you some of the differences we're seeing. On the left, TDCPP, that's that chlorinated flame retardant chemical. So in the 6 individuals that indicated they came across a structure, their exposure to TDCPP was significantly higher. It's around 5 times, 5 times higher on average versus the wildland firefighters that responded to were working in areas that it was really just more timber and shrub and grass that were on fire. Phenanthrene showed the same pattern. This is also one of those combustion byproducts. This was about 5.6 times higher on average. Again, a small number, but clearly shows these differences. And this probably makes sense because once you have a structure, there's more fuel load, generating more of these chemicals that are probably in the air and sticking to those wristbands. So then we can clearly see these differences when we make these measurements. Another question and something that's been of interest, I think, to the fire service is understanding the implications of gear washing, right? Or washing our PPE, or even sometimes the type of PPE we wear. So one of the survey questions that was answered was your PPE laundered prior to this exposure? So when they put that wristband on and went out for that shift, was that a clean set of PPE, or had it just been washed? Yes or no. So this is a yes or no answer. So of the wristbands that we analyzed, 15 of those wildland firefighters said yes, this was freshly laundered. 38 said no. And so here I'm just showing some more of the chemicals because most chemicals followed a very similar pattern to what you see here, such that if they indicated yes, that it had been freshly laundered, those chemicals were on average lower on the wristbands, many of them significant. These ones that I'm showing you here were all statistically significant. In the case of that chlorinated flame retardant TDCPP, it was about 3.7 times higher on wristbands if they said no to that question. The combustion byproducts were on average about 3 times higher on wristbands. And DBDPE, this one in the middle, that's a brominated flame retardant. That was about 1.6 times higher. But there were 16 chemicals in our panel that showed this same pattern, 16 that were commonly detected, reinforcing that these chemicals that are emitted from the soot, to the fire and the smoke, they stick to that PPE. And even when we're not near the fire, it seems likely that it can be continuing to lead to exposure through inhalation or dermal exposure, and reinforces the importance of washing that to reduce that exposure, so that when you're away from that fire, we probably think that there's no more exposure, but there could still be continued exposure while wearing that PPE. And so important to think about that in terms of mitigating these exposures and risks. So some of the takeaways here, I hope this has convinced you that there's some utility in using wristbands to help answer some of these questions about chemical exposures. I think they can help us understand variables that are influencing our overall exposure. And certainly we're looking for other ways we can use these to help ask and answer questions of the fire service. So if you have suggestions, please send them out. Here we did see that the levels of combustion byproducts on the wristbands were significantly correlated with levels measured in paired urine sample. Again, reinforcing that they're measuring meaningful exposures. In the future, we're hoping to look at some of these other flame retardants and plastic additives in urine to understand and just verify that they are also correlated. Again, reinforcing their use. We see clearly signs that job tasks influence the chemical exposures that wildland firefighters are experiencing. We are trying to apply this to some of our other fire service partners and groups too, to ask and answer this question, because it's important to determine if these do replicate in other studies. And lastly, so clearly there's some evidence that laundering and washing our gear can help reduce our exposure, not just to combustion byproducts, but other things as well, because there's other things that are being emitted from these fires, particularly structural fires. So it's not always just combustion byproducts. It could be a lot of these plastic additives as well. And washing your gear as frequently as possible can help reduce that exposure. Now we're continuing to analyze wristbands for other studies within the fire service, and I just wanted to kind of give you a sense of what else we're working on right now and what other directions we're hoping to go. We do have some wristbands that were worn by first responders from the LA conflagration that occurred about a year ago. We had 17 of these that was sent to my lab for analysis. I have the data. We got a second set recently, and we are working on merging those data to perform the statistical analyses. I will share that the preliminary findings from that first set of wristbands from the LA conflagration, we saw the exact same trend based on how the responder indicated whether they had worn freshly laundered gear or not was statistically significant. We are continuing to look at that and are seeing some differences based on fuel sources. We have more wristbands that are coming in from wildland firefighters, and we want to look at some of these exposures, how they differ on duty versus off duty among wildland firefighters. We have some wristbands from firefighters in New York City that were responding to fires associated with e-bikes and e-scooters. I know there's lots of questions about what type of exposures first responders are experiencing during these fire events, and we're hoping to look into that a little bit here shortly. We're also deploying wristbands in the North Carolina firefighter cohort here. I'm particularly interested in asking questions about how long you wear your turnout gear and what type of turnout gear. Is this influencing exposure to PFAS and some of these brominated flame retardants? And we're additionally looking at how cleaning frequency relates to exposure. And I think in the future, I'm hoping that we can look at exposures over time. How do our exposures change, not even from fire to fire, but from season to season and year from year? I think this can be really helpful for understanding some of these risks. So with that, I want to make sure I can give lots of time for questions if there are some. And I just want to thank, this is a team effort. Certainly my lab helps with these analyses, but this is, partnerships with multiple fire service partners, particularly Jeff's group at Arizona. Thank you for all your help and assistance. I want to certainly thank our partners here in North Carolina and our funders, and my good colleagues down at North Carolina State University, who we work with very closely as well. So I will end there, and I am happy to take some questions if there are questions.
Sure, Heather. Thanks. And you can feel free to call on individuals like Derek Bering, who's got his hand raised, and we can go on.
Okay. Well, let's start with Derek then. I'll open up the chat. Derek, do you have a question?
Yeah, Heather, thank you so much for all this. I learned a ton today, so I appreciate it.
Sure.
The question I know you're talking about, the value of cleaning PPE, and this for us, like cleaning it with an extractor is our turnouts and hood, but the onus falls on the firefighters to clean your own gloves and boots and helmet. And so, I just don't know if we're actually getting the exposures off of those things by not putting them through an extractor, which you can't really. But what do you think about that? And is there better ways to clean that stuff or certain cleaning products? Because, our department's saying just soap and water is going to get carcinogens off of gloves or off of helmets. And do you believe that to be adequate enough? Or do you feel like we should be doing a deeper dive of how to clean those other products that aren't put through an extractor?
Right, yeah, that's a great question, Derek. I have two thoughts there. Well, first, I think it may be helpful for us to think about asking those types of questions in future wristband studies. Maybe we can collect information on what exactly is being washed and under what frequency, so we can differentiate how is it important it is to wash either just the turnout gear, or how important is the hood versus the gloves. We haven't really been framing our questions in a way to answer that, and maybe in the future we can do that to get to that question. I think that's important. On a separate note, I have some collaborations here with NC State and some colleagues at Duke where we've been trying to ask this question about washing of the turnout gear, focusing on jackets in terms of how much is actually removed, because there's certainly studies out there and research done. Most are on PAHs. So if we're just looking at PAHs, it's actually a very narrow window of chemical space. And given their solubility in water, it's not surprising we see them come off pretty well with water and soap. But when we look at things like brominated flame retardants, which I know firefighters have exposure to, they have different chemical properties. And our data seems to suggest that they don't come out of the gear during standard cleaning as well as pHs do. Like we might get 80% of the pHs coming off gear when you wash it, but maybe only 20% of the brominated flame retardants. And I think we should be looking at in the future, a good question to ask to understand that. Maybe it's not coming out, but does that mean you're going to get dermal absorption to that? I don't think we know. Does it just stay in the gear? Does it pass through the skin? To me, I think these are things we don't know the answer to, and we should be focusing on the future. So I'm not sure that's answers your questions, but I think it's important that we think about those as we conduct further studies moving forward.
Awesome. Thank you so much.
Welcome. Trying to go over to the, oh, sorry, Melissa, you have a question. You have a hand up?
Yeah,
Thanks, Heather.
sorry.
That was
Go
a
ahead.
really nice presentation and overview of a lot of recent work, I guess, that I haven't seen before. So it was exciting to see. So for the correlations between the the urine and the blood and the wristbands, I was kind of surprised that they were so high because you're not capturing any sources from food or from water, which I think have traditionally sort of been hailed as the predominant sources of PFAS. So I guess I'm surprised that it seems mostly to be from dermal or from air. And I just wondered if you could comment on that a little bit more.
Yeah, no, great question. I didn't know how much to get into that on that side. So it's important to note that the one correlation I showed you was for methylphoce, which breaks down into N-methylphosa, which is one of the NASEM 7 where there's concerns about health. My feeling, based on all the data we've seen, because we have replicated that in a cohort in Michigan now, is that methylphoce exposure that breaks down to N-methylphosa, that's really the indoor environment. I think it's all inhalation or dermal. It's hard to differentiate exactly how much is inhalation versus dermal, but it's the indoor environment. I don't think there's a lot of exposure to N-methylphosa from our diet, unless it's really different in North Carolina and Michigan versus other areas, which it could be. But things like PFOA and PFAS and these others, I definitely think diet is main, because we've looked at those on the wristbands. We see no correlation whatsoever for PFOA and PFAS, PFHXS, these other common PFAS. It's only N-methylphosa, which is strongly correlated with methylphoce.
Okay.
So I think there's some PFAS in which the indoor environment plays a big role. In this case, the N-methylphosa, but others, I think it's really all diet.
Okay, and this might be too technical of a question for this audience, but are there specific properties of N-methylphoce that make it more susceptible to inhalation exposures?
Well, N-methylphoce is what we call a neutral PFAS. It doesn't have a net charge on it, so it's more capable of migrating out of these products and entering the indoor air and indoor dust particles, where you can lead to exposure, which is different than PFOA and PFAS, which are ionic and can be charged. So their state is a little bit different in the environment. So that's why we focus a lot on these more neutral volatile PFAS in applications with the wristbands.
Okay.
Thanks,
Heather.
Sure. Okay, I opened up the chat because I know there were some questions. Hopefully I got to the top here. I see one that says, have fire investigators participated in the study? In the studies I've talked about so far, we have not had any fire investigators included, but I certainly agree that's very important. And I'm hopeful we can do that in the future. I don't know, Jeff, if you can comment on whether any fire investigators have been involved in the other projects we have ongoing where we're deploying wristbands.
Yeah, we actually, NIOSH was kind of in charge of the expansion grant, which was run by Alberto Caban Martinez. And so he should have some information, but I don't know that they did wristbands, but I think they were doing exposure monitoring for the fire investigators. So there should be some information forthcoming there.
Right. We're certainly open to it in the future. I think that would be a great application as well. I see a comment from Sean. It would be interesting to use these to look at effectiveness of clean cab strategies being implemented by departments. Since movement is important for the wristband to aid in absorption, would this still be a useful tool despite the movement challenge, not on an individual? I certainly have thought about that question. We actually have talked to the Durham firefighters about deploying wristbands as a passive air monitor inside the cabs of some of the rigs, the fire trucks. We are thinking about doing that. It's just important to know, I think that can certainly be done. They can be used in applications, but when you interpret the data, you have to be a little bit careful because we do know movement influences the rate at which these chemicals accumulate. So I would be hesitant to compare data from a wristband used as a stationary air monitor with ones that are worn by people. You are going to see differences, but they can be used. You just got to use caution in how you compare them. But I do think that would be an excellent idea and something I have thought about as well. So hopefully that
Yeah, thank
answers
you.
your questions. You're welcome. Have any tests evaluated the products from lithium-ion events? I know we have some projects ongoing where we're trying to answer that. I also have a collaboration with FSRI where they are under controlled conditions in a laboratory, burning some of these batteries, lithium-ion batteries, to look at emissions. So I hope we'll have some data on that very shortly. So I won't comment on any more on that. I see a question from Jeff Hughes. Is there an ability to check for BFRs in the blood of our firefighters during annual follow-ups? Great question. I put in a grant to do that exactly. It actually got a really good score on the first round of review, but was not funded, but we have put it back in. It'll get scored in April. I'm really hoping we can look at that in the future. Those studies just unfortunately are really expensive. So we have applied to NIH to get some additional funding to support that question. So stay tuned. I'm hoping we'll learn more. I see another question here. How strong is the adhesion of volatile PFAS to silicone wristbands? Since this is the kind of passive sampling, as different firefighters face different work, will the environmental factors affect the absorption of PFAS? That's also a really good question. We do know that based on things like vapor pressure and octanol air coefficients, how stable they can be on the wristbands at certain temperatures. Right now, they've only been tested up to 35 degrees Celsius, and they are stable over that period. I agree this is something that should be looked at in terms of higher temperatures, and something we should look at in the future. Once you start getting some particles and things stuck to the band, they're less likely to volatilize off. So I think I'm hoping that should be rather limited, but it is a question that we should ask and answer in the future. We have done some studies where we've had people wear wristbands, start with 5 and take 1 off each day. So we can look at accumulation over time. How stable are they? Do they keep going up, or do they reach a plateau? For everything we've looked at so far, they do actually go up with time, but PFAS was not included in that initial study. So we are working on repeating that so we can assess that question and answer that. So another thing to stay tuned for. Jeff Polly, I see your hands here. Thanks for joining, Jeff.
Of course, of course. So there was the only, going back to the question about wristbands and fire investigators, the only study that's been done, very limited, here in North Carolina with University of Miami folks, about 35 people submitted wristbands. I'm in the process of looking for, I've got the study. I'm going to put the name of it in the chat so people can reach out and find it. It'll take me just a minute, but that's the only fire investigator wristband study. So we could use more.
Yes. Agreed.
Just saying. Thank you, Heather. Great job.
No, you're welcome. Thanks for being here. Well, hopefully we can figure out a way to do that in the future. I agree. It's something we should look at. Judith.
They have a fantastic body of work. Thank you so much. It's really exciting to see these results. I'm wondering, in terms of the levels you're seeing, I mean, granted, any exposure needs to be reduced, but I'm wondering where these stand around what's known around health impacts. As you said, a lot of these chemicals we don't know, like a lot of the PFAS, we have no idea if they impact health because they just haven't been studied. But I'm wondering if you're looking at it or what your plans are to look at it that way. Thanks so much.
Yeah, no, that's a great question. I think there are a lot of questions when using wristband about how to use the data in a risk and health framework, right? And so this is something a lot of us are wrestling with. And right now there's 2 schools of thought. I think there's one thought where you take a wristband measurement and using calibration studies, you back calculate what the air concentration is, and that relates to what you're inhaling and your inhalation risk. I am not a fan of that for reasons I mentioned earlier, because we know movement impacts this in quite a bit. We actually published a paper on this earlier, where just walking speeds can increase the uptake rates by up to 5-fold, 5 times higher uptake rates based on just walking. So I think what we need to do in the future is look at more of these correlations with internal dose, what's in urine, what's in blood, and work on using machine learning algorithms to say, okay, here's your wristband measurement. So with some
The
degree of
harness is through.
uncertainty, what's the expected level in blood? And then can we put that into the framework? But you're right, most of the PFAS that we pick up on the wristband are these neutral, more volatile PFAS that don't have a lot known about the health impacts. Although in the case of methylphosph, I should point out methylphosph breaks down to N-methylphosph, but can further break down to PFOS, which is a concern. It's just my sense is most of the PFOS is from diet, and there's small fraction coming from the further degradation of N-methylphosph. These acronyms are a nightmare even for chemists like us. I will just point that out.
Thanks so much.
So
Any other questions? I'm not seeing any other questions here.
no, I just was going to give folks just a moment to see. If they have anything, they could raise their hands. That was a fantastic presentation, Heather. Really nice, nicely laid out, easily understood. So, yeah, there we got Brett's hand up. I just wanted to really echo your comments about the laundering of the gear. So, I mean, this is something was particular, I think, to wildland. It's a little bit harder logistically to do it, but it is something that I think we have evidence base for now. And we're finding similar things in the urine of the firefighters. So that's something that we shared with them as well, too, that backs up or actually it came, just for everyone to know, the original observation came from Heather, and then we looked at it in the urine and we were finding similar effects with the PAHs. So this is something that's, we're already shared with the participants, and we're hoping to get this submitted for publication so we can share it more broadly. So particularly in the wildland world, wearing clean gear whenever possible, I think will be an evidence-based intervention. So with that, I'll pass it off to Brett.
Hey there. Yeah, just following up on a comment I submitted talking about the clean cabs area study, and just an additional question on if there's been any evaluation of the SCBAs themselves. I know that there's there are extractors for them, but they're very far and few between. And so the typical decon method for an SBA is to spray it off, maybe with an air hose, maybe with some water, maybe some soap, or maybe people are just leaving it in a cab dirty, which they shouldn't do. But that I've seen that happen more than once. And so just the evaluation of that exposure risk in and of itself, just sitting in the cab with that off gassing still, and some measures that we can take to provide that level of exposure risk reduction if we don't have a high-end SCBA extractor.
Right. Oh, that's an excellent comment. I see if we can revisit these discussions about deploying some of these in the cabs of the trucks and the rigs, because I do think that's something important that maybe we should be looking at further. And if we were to do that study, then we could probably take note of what the items are in there, or things that have come in and out of the cab while the air sampler was deployed, to try to ask and answer some of these questions. So yeah, thank you for that comment. And I'll just add that if there are other suggestions, other questions you have where you think wristbands can help answer them, please feel free to send them to me. It's really easy to reach me. It's just my name, heather.stapleton@duke.edu, because I think we are hoping we can maintain some funding to keep the research going with wristbands and here in North Carolina. And so I'm happy to apply them in the best way possible to answer questions that are important to you.
Thank you, and I especially appreciate the detail to the study, having the wristbands for off duty too, because I know that that's another area of potential exposure. So that was great to have that. And then it was mentioned briefly in one of the earlier slides, and we didn't really talk about it today. But has there been an evaluation into sleep disruption itself as the cancer causing agent, aside from the chemical exposure, a way to impact that at all?
Sure. So I think the audio was going out a little bit, but I believe the question was, is there any research investigating sleep activity or sleep disruption as a risk factor for cancer separate from chemical exposures? And yes, there's research that's ongoing and looking at that for sure. We recognize that as also an important stressor that occurs in the fire service. I'm not as familiar with some of the ongoing studies in that area. Jeff, maybe you know more or you want to add anything? Jeff Burgess.
Sure. I mean, we're using some of our epigenetic analyses, and I think at least more recently, I have to look how far back, we had information on extent of sleep, particularly off shift. And so this is something that we'd be able to address. But yeah, I think even just more broadly, as Heather has mentioned, there's a lot of information about how important sleep is for almost everything. So it's something that we'll be continuing to look at, but also there's a lot of information out there as well. And the more sleep you can get, the better you are for sure.
Yeah, great. Derek Bering, I see your hand up.
Yeah, you just made a comment a few minutes ago about you feel like PFOS exposure is from diet. I haven't heard that yet, so I wasn't sure what your feelings are with that.
About PFAS exposure in general or specific to firefighters?
I don't know. You just made a comment a couple minutes ago about you feel like PFAS exposure is more due to diet. And so
Yeah,
wasn't sure what you meant by that.
sure. Let me comment on it. I know it's a complicated. There's the term PFAS for the whole group, and then there's specific PFAS, which the acronyms sometimes get a little confusing too. I think we know in general that diet plays in the general population that diet plays a strong role in our overall exposure to PFAS, particularly PFOS and PFOA, which are some of where we get most concerned about health impacts. Within the fire service, we do know there can be additional exposures from the use of AFFF, particularly for PFOS and PFHXS, which were dominant in some of those AFFF formulations. And then there's certainly the concern and questions about exposure from the turnout gear that has been treated with PFAS in the outer shell and the moisture barrier. I think certainly there's data that suggests that firefighters, based on blood measurements, on average, do have higher exposure to a few of those PFAS. So some of that we do think is from AFFF. Some of that could be from the things that stick to the gear and lead to exposure. I don't see any evidence, at least in North Carolina, that it's from release from the turnout gear, from PFAS treatments in the turnout gear. Right now, for the ones we're concerned about for health outcomes like PFOS and PFOA and PFHXS, we just don't see that yet. Certainly, it's an area that needs more study. But I think in all departments, there's going to be challenges in trying to understand how much is coming from drinking water contamination or diet versus what came from the station or in the air. I mean, we do have some data suggesting PFAS are in the air in fire stations. So there's some inhalation exposure going on in stations, and it's quite variable. It can be variable from one room to the other in the fire station. So there's lots of questions that we have to ask. But in general, we know that diet is a main source of exposure for PFAS across the board. It's just looking in the fire service, how much additional comes from these specific areas like the use of AFFF and the turnout gear. And that's going to vary from one type of PFAS to the other.
So by diet, do you mean like the cooking items you use to cook with, or do you mean specific kinds of foods and stuff like that that are affecting the PFOS?
Right. Well, we certainly know that some areas have contamination in their drinking water, right? And they're working on remediating water, treating water. But then we know that produce that's irrigated with contaminated water can have PFAS in there. And then there's the issue of food packaging. It's clear that there are PFAS that have been used in the packaging, in the plastic films, and in some paper for food products, and that has led to migration into food. So there are food products that contain PFAS. Microwave popcorn got a lot of attention a long time ago because the paper used in the microwave popcorn packages, and when you put it in the microwave, it would leach out into the popcorn, and then there was exposure through that route. But it's been changing a lot over time. A lot of manufacturers are phasing them out, so it's very dynamic and it's changing over time. So it's very hard to tease these out, is the point. So in general, diet plays a role, but it's not the only role. As I mentioned, there is this one PFAS where it clearly looks like inhalation indoors leads to exposure, probably from what's being released from stain repellent upholstery and or rugs or carpets, things like that.
Awesome, thanks.
Okay, with that, I think we've reached the end of the hour. Again, Heather, thank you very much. Excellent presentation. I just put something in there on the chat that Heather is willing to take additional questions from you directly, and that we'll share the video on the website. So thanks again, everyone, and we expect to have another episode of this in about 4 months, and we'll let you know beforehand. Thanks again. Have a good day.
Thanks, everyone. Bye.